Headlines, Life in a Test Tube, exclamation point. I shall never forget the headlines, the emphasis, the discussion in the classroom, 1952 and published in 1953 in the Technical Journal. The experiment of Stanley Miller, commonly called the Miller-Urey experiment because Harold Urey was the mentor and supervisor of the laboratory and supervisor of the experiment that took place. An apparatus was set up, and the four basic supposed components of the atmosphere in early historic times of primitive planet Earth were sent through a series of test tubes and a trap, a spark chamber, etc., etc. I shall never forget the discussion in class. I was in my middle years of school at that time. I shall never forget the statement made by the teacher. They have now been able to synthesize life in a test tube. We go beyond that, and the headlines over the years have stated, well, they've already synthesized life in a test tube, so the progression of living systems from that synthesized life will prove to be less than complex, with a smile and on to the next article. It's always amazed me. The time of this publication, that was almost 60 years ago. Since that earth-shaking headline, there has been no further progress toward living systems in a test tube. Did they really synthesize life in a test tube? What are the real facts? Before the program is over, we're going to hear the published statement of the supervisor of the project. The problems are insurmountable, and we'll report at the end of the program whether life was synthesized in a test tube and what the possibilities really are. I have two special guests today that I welcome to the program. This is a global communication forum. Doug Sharp, you and I have been friends for a long time.
Yes.
You've excavated with me along the banks of the Biloxi.
That was a privilege.
You have your own program. You've produced over 400 telecasts yourself. In addition to that, you've written numerous books that have been published internationally, etc.
Yes, we have had the opportunity to do that for quite a number of years. Since 1993 was the first time we started producing our own show, Revolution Against Evolution. And
all right.
That's been a lot of fun.
A lot of fun, and you've interviewed a lot of world-class scholars, and you're a very fine scientific scholar yourself with a background in chemistry, math, physics, and the ability to synthesize in a computerized format what you can envision chemically. We're going to talk about that later, which is the main thrust of the program. But I want to step internationally now. My dear friend and brother, Jyoti
Chakravarti.
You are the president, CEO of the Creation Science Association of India.
That's right.
You founded the movement in India, I understand.
That's right. Maybe the only creationist group surviving in South Asia.
That's marvelous. I commend you. And you came to the United States recently to lecture in a major forum at a very major symposium, and I commend you for your research. Now, I understand that originally when you heard about, you were much younger than I when you first heard about it, but in the university and college classroom, did you hear about the Stanley Miller or Miller-Urey experiment, life in a test tube?
Yes, because that was the same time India was testing with the test tube babies. So, you know, there was thinking of almost, we were thinking that the next stop forward is going to form a life in the test tube.
Great. That states it very well. So, what happened in India?
Well, when being raised up in a Christian home, I was forced to believe in the Bible. But then when I went into college, I went on with all the scientific details and research. But then I felt, okay, God created, it took billions of years. But one thing struck to me was, it's a long story, can we create blood? in laboratory.
Which is absolutely necessary for complicated living systems. And the answer was?
Still no. I said, you will never be. And I found there must be a creator and there must be a process which we still can't put in our laboratory.
In fact, the process not only must be orchestrated by intelligence imposing his knowledge, his skill upon the chemical elements, but it must be done very rapidly.
That's
otherwise it all biodegrades and you end up with nothing. Now, at that time, you were a creationist because it was imposed upon you. And then the announcement came, and in class you were taught that life had been synthesized in a test tube. So you assumed that it was true, and so you adopted theistic evolution or progressive creation. It's really same concept, different sides of the coin.
Yeah, it's another form of evolution. But the question is, even if you form a life in a test tube, you need a womb.
Certainly, and all must be intact simultaneously.
Yeah.
But since then, you have seen that life was not synthesized in a test tube, and you have found that even progressive creation does not address the issues. It simply accommodates theistic concept, evolutionary concept.
That's right.
It is not satisfying. It is not truthful. In fact, today we're going to get to the truth of the matter. Thank you
very much,
Jyoti.
Dr.
It's a
Brown.
pleasure to have you.
Thank you.
Now, Doug, let's get to your specialty. I'm going to step beyond these gentlemen to this side of the placards, and we want to succinctly and rapidly carry you through the progress. Has life been created or synthesized in a test tube? Doug, it's such a pleasure to have you here, and with your background and your ability to analyze, synthesize, and write. In fact, you work for governmental agencies in computer synthesization in the Natural Resources Program. So you're a recognized authority in this area.
Yeah, I write computer applications for the Department of Natural Resources. So I have on my website a computer simulation of the idea that you can, by random chance, produce a meaningful sentence. And this was Dawkins' methinks it's like a weasel thing. And I point out what the assumptions he has to make in order to do that, and demonstrate if you change these assumptions and make it more like reality, then his hypothesis breaks down.
Breaks down completely, like the amino acids in the test tube, as we're going to see.
Yes.
Now here we have an illustration of evolution's imaginary road to life. And there was a time you and I both in our earlier young career espoused or at least embraced and entertained evolutionary theory. And according to evolutionary theory, and I think this will introduce the concept to the audience very well, here we have hydrogen cyanide, ammonia, formaldehyde, carbon monoxide, and water, which were the essential ingredients used by Stanley Miller. And you have formation of a purine base, and hopefully then you'll get some sugars and fatty acids and nucleotides and amino acids, and hopefully that will then self-organize into a protein, into the DNA, add the lipids or fatty acids, and produce a cell. Wow, here we've solved the problem, life in a test tube. But as we're going to learn, this occurred nowhere but in the mind, the hopeful mind of the evolutionists. In fact, it was stopped dead in its tracks with the Stanley Miller experiment. And Doug, please explain briefly Stanley Miller and his supervisor, Harold Urey.
Well, Stanley Miller was given the idea that he wanted to produce a thesis, graduate thesis, and actually Dr. Urey encouraged him to do something a little bit more pedestrian, something a little bit more ordinary, because this particular idea that Miller came up with to try was something that had a high chance of failure. Well, it failed anyway, but he put together a scenario where it produced the illusion that you're producing life in a test tube.
The illusion. And you and I bought it for a while.
Oh, yes, and my interest in this came about because I had built a Van de Graaff generator. It stood about this tall. It had a big dome on the top. I had a 5-horsepower motor on the bottom, and it had a belt that ran rapidly up and down, and it would pile the electrons on top of this dome and shoot off sparks about 6 feet long. And so that terrorized my parents and the neighborhood. And
well, then how old were you when you actually?
I was about 17 when I built.
17 when you built a Van de Graaff generator.
And so I entered this in a science fair, and next to my exhibit was another fellow who had won the grand prize, and his exhibit was the Miller-Urey experiment. And he also had a Van de Graaff generator that powered his spark apparatus, like what Dr. Miller has here. And so that really piqued my interest. I said, wow, life in a test tube, this is what it's all about. I want to investigate this further. And I was taking a class at Lansing Community College, which was for High School Honors Institute, and they allowed us to actually do real science. Good. Just something that, to actually put together a hypothesis, test it, and try to see if that hypothesis is correct. My hypothesis was that if Stanley Miller can produce these amino acids in the test tube and be able to make them available to produce proteins, well, the next step, of course, is to combine them together to form proteins.
Yes, hopeful step. Not demonstrated, but hopeful in this process. Okay.
So that's exactly what I attempted to do, and what I found out was, well, I had made some mistakes in my assumption. I had my amino acids that I picked out, and then I picked out alanine, glycine, tryptophan, and tyrosine, which were the only ones that they actually had in their laboratory for me to use. And so I said, well, we'll try these and see if we can get the peptide bonds to work. And but my assumptions with it was that you have a warm pond in which these are going to be sort of fermenting and combining together to produce that. And so what we ended up doing with it was I jeopardized my experiment because I put it in solution. I found out later that putting them in water is contrary to what I needed to do to actually perform the peptide bond. And so I needed to have them somehow out of solution. And that made a problem for me.
It was an impasse.
An impasse. It just didn't work. The second thing I found out was that these chemicals that I got off the shelf in the laboratory were racemic. And what that means is that they had 2 different isomers mixed together. But life only uses the left-handed kind.
Left-handed kind.
And you have left-handed and right-handed amino acids
that form naturally.
That form naturally.
That formed in his experiment.
That formed in his experiment.
But that's not exposed in the propaganda.
Right. And we're going to be talking about this a little bit later because Dr. Urey was asked this question.
That will be a climax. To let you in on what he's talking about, here he reran the same experiment in its basic form. He was able to get amino acids, but amino acids are not all you need. You need the formation of nucleotides and sugars and purine base and fatty acids to get to protein and then to DNA and lipids and cell.
And it turns out the DNA needs right-hand sugars, and the lipids need a cis conformation rather than a trans conformation. You've heard of trans fatty acids.
Yes.
And these are all conformations that life has and needs to produce, but you cannot form them naturally. You get equal mixtures of both.
So, Doug, has life been created or synthesized in the test tube?
You haven't gotten even to the beginning before you get some problems.
Major problems. In fact, since we need to get all of this in the program, let's emphasize this. You need the right materials of the right kind, left-handed amino acids, but they come in nature when they're formed, and in his experiment, right and left-handed. Right hand won't work. It takes life to extract and break down the right. You've got to have life before you can run the experiment. Isn't that amazing?
That certainly is true. You need it in the right place, the right time, in the right environment, in the right quantity, in the right concentration, in the right order, in the right staying power is what they can't break down and fall apart.
No, as they do in nature. All right, here we have a diagram of this system. You have the standard ingredient materials. You have your accelerator, your spark chamber, but he used a trap. He didn't let it recycle and bio-degenerate like it does in nature, so he fudged the situation. Now let's quickly go to the other side, and here we have an illustration of life originating, or simply assumed to have originated and produced the mind of man that can look back and see how it happened. But there's no evidence that it happened whatsoever. Let's go to the other side of your computer simulations, and then we'll get to Mr. Urey's specific statement that I want this audience to know about. The left-hand amino acid dilemma. Here, describe this in brief for us, please, Doug.
Okay, this is the left-handed alanine, which is one of the amino acids, and it's used by life. This is the right-handed alanine, which is rejected by life.
But in the test tube, you get both of those. No life can be formed from this.
And what is interesting is that there has been some proteins that have been artificially synthesized, and I believe it was Barry Commoner that tried this, where you had both kinds of amino acids in this protein. Well, he put them in a living system, and the living system took them apart and only used the left-handed amino acids out of it.
So to synthesize even a protein, you need a living system to perform it. So you can't get life from it to be non-life to produce it. As if
there's a divine barrier here, or a divine force that is going in the living system that is selecting out exactly what is needed to produce the right materials.
By very intelligent design.
Exactly.
Not only have we emphasized that you have a background in chemistry and math and physics and are recognized as such, but in your computer simulations, you've been able to demonstrate the tail of a snail. Would you describe that for us, please?
Okay, well, what I did was I took the odds against, which is published in Coppedge's book, Evolution is Possible or Impossible. And he says that the odds against a normal protein of average length forming by chance is 1 chance out of 10 to the 123rd power, which is being somewhat generous to the whole thing. And now what I did was I took that figure and I said, how can we actually explain this concept so somebody can actually understand it? What it is that we're giving the snail a task to do. His task is to move the entire Earth to the other side of the universe and back, one molecule at a time. Furthermore, the snail is a very slow snail. He can only move 1 millimeter in a million years. Okay, so he's going to take a little while to get this done, okay?
To move the entire Earth to the other side of the universe.
Now, suppose you're the engineer and you have a protein factory that produces 400 tons of proteins a second you're really cranking out the proteins, okay, with left-handed and right-handed amino acids.
400 tons of proteins already. You've jumped over the impasse, the barrier. You've granted the proteins, and you're cranking these out at 400 tons a second.
And so here's the issue: is that our protein plant will have filled the entire universe with proteins, none of which are viable, by the way, before 1 possibly one is viable, and still the snail will have won the race in producing, moving the entire Earth to the other side of the universe and back.
Oh my! Now this communicates to the audience. Here you've jumped over the impasse of these left and right-handed amino acids, and you've granted to the evolutionists that, okay, you can somehow produce proteins, okay, but they have to be viable. So to get one viable protein with which to start a spark of life, but you need a lot more than protein to start the spark of life, he produces, you give him a factory producing 400 tons of proteins per second, and he can fill the universe with these proteins to get one viable protein, but the snail moving, how fast?
1 millimeter in a million years.
In a million years, to move the Earth to the other side of the universe. But somehow, according to the mathematical odds, you say, and it's been demonstrated, that the snail will move the Earth to the other side of the universe 1 millimeter each grain, each molecule.
The figure that was given, and this is a very generous figure that he has given to it.
He would win the race against producing one single viable protein. Doug, you're telling me that Stanley Miller and Harold Urey did not produce life in a test tube.
They produced a few amino acids.
Which is not life.
Racemic amino acids, of which I had in my off-the-shelf chemicals.
Left and right-handed, which do not work in life.
That's
life
right.
only uses the left-handed. Now, in these closing moments, I'd like for you to read the direct statement of Harold Urey decades after this experiment, which he and Stanley Miller were purported to have created life in a test tube. Have they? Would you read his direct statement in a confrontation published?
Okay, there was a symposium that was done in Lansing where Harold Urey was invited, and he had a question and answer period after his presentation, which, by the way, the arguments in it seem quite dated now, but I thought it was interesting. But here's what the question was. In the biochemical scheme for the formation of life, I guess people have worked under you, have developed or synthesized amino acids and some proteinide substances. Characteristics of these substances is the fact that they're always isomer mixtures. All life is characteristically optically active or made up of optically active compounds. How would you account for the formation of optically active biochemicals from racemic mixtures by chance?
Left and right-handed.
And here's what he says. Well, I have worried about that a great deal, and it is a very important question, and I don't know the answer to it. Characteristically, as I say, I tell my scientific friends to go back and work on it, and maybe 10 years from now, maybe 100 years from now, we will understand it better than we do now. You won't understand it any better by simply by saying that God did it. You will still be saying that 10 years from now, and you will find many people who will puzzle about it, but I would make this suggestion. And he just goes on and
just
punts on
withers
this
away.
punts on this whole question.
That's when I asked Doug Sharp about this. He stated, well, what he really did was punt on the whole question. Doug was involved in writing articles in the production, stage production of Inherit the Wind. Now, as we conclude this material, Inherit the Wind, of course, ultimately states there's real credence to evolutionary hypothesis. The Stanley Miller experiment states not only is there credence to evolutionary hypothesis, the building blocks of life have actually been created naturalistically in the test tube. But we found on today's program, what they created are amino acids that are, some of which are actually found in the polycarbonates out in space. That takes us nowhere. They're racemistic. That is, they're left and right-handed, but life only uses left-handed amino acids in the building as the building block of life, but then you get a viable protein beyond that. Beyond the protein, you must get 60,000 of them in a specific configuration to inhabit one functioning cell. Was life created at the University of Chicago in 1952 in the Miller-Urey experiment? No. The real facts, before the program is over, we're going to hear the published statement of the supervisor of the project. The problems are insurmountable, and we'll report at the end of the program whether life was synthesized in a test tube and what the possibilities really are. I have two special guests today that I welcome to the program. This is a global communication forum. Doug Sharp, you and I have been friends for a long time.
Yes.
You've excavated with me along the banks of the Biloxi.
That was a privilege.
You have your own programs, have stated, well, they've already synthesized life in a test tube. So the progression of living systems from that synthesized life will prove to be less than complex, with a smile and on to the next article. It's always amazed me. The time of this publication, it was almost 60 years ago. Since that earth-shaking headline, there has been no further progress toward living systems in a test tube. Did they really synthesize life in a test tube? What are the headlines? Life in a test tube! Exclamation point. I shall never forget the headlines, the emphasis, the discussion in the classroom, 1952 and published in 1953 in the Technical Journal. The experiment of Stanley Miller, commonly called the Miller-Urey experiment because Harold Urey was the mentor and supervisor of the laboratory and supervisor of the experiment that took place. An apparatus was set up, and the four basic you've produced over 400 telecasts yourself. In addition to that, you've written numerous books that have been published internationally, etc.
Yes, we have had the opportunity to do that for quite a number of years. Since 1993 was the first time we started producing our own show, Revolution Against Evolution. And
all right.
That's been a lot of fun.
A lot of fun, and you've interviewed a lot of world-class scholars, and you're a very fine scientific scholar yourself with a background in chemistry, math, physics, and the ability to synthesize components of the atmosphere in early historic times of primitive planet Earth were sent through a series of test tubes and a trap, a spark chamber, etc., etc. I shall never forget the discussion in class. I was in my middle years of school at that time. I shall never forget the statement made by the teacher. They have now been able to synthesize life in a test tube. We go beyond that in the headlines over the years.